Hi everyone, my name is Ella Weldy. I'm a master's student at Clarkson University studying under Dr. Grimberg. Today I'll be presenting my research on reducing fugitive emissions from manure lagoons for the 2026 Center of Excellence Conference and lightning talks. So as we know, climate change is a major pressing issue right now, and methane as a potent greenhouse gas is a priority to reduce the emissions of. Manure lagoons and manure in general is a major source of methane emissions, specifically in agriculture. In fact, about 10% of greenhouse gas emissions from the agricultural sector come from manure lagoons. We were looking at bromoform for this particular study because bromoform has been found in the seaweed Asparagopsis taxiformis and has been used to decrease enteric methane production within cows. So it's fed to them as a supplement. The bromoform is the active ingredient and it's been found to effectively reduce enteric methane production. The promise of this research is then expanding that finding to include manure lagoons, recognizing that they're also a major source of methane emissions. So the question in this research is, how does bromoform impact methane production over a month-long period? Importantly, why? How does that work? To do this, we used 3 different experimental groups of varying concentrations with a control in blank and analyzed their methane production over 40 days. Methane production was analyzed using an AMPDS-3 system, it's a form of biomethane potential analysis, in which bromoform, manure, and digester effluent were confined in a reactor bottle. A carbon dioxide scrub was used, and then gas production was measured in a flow cell. Finally, an organic solvent was used to remove any residual organics from the gas before it was vented to the atmosphere. This graph shows us the overall results of methane production. From this, we see that there's an overall almost complete suppression of methane production for about the first 15 days, at which point we have a breakthrough event. And for the following 20 days, we see a pretty substantial increase in methane production in that 200 micromolar group that was the highest dose that we looked at. This led us to ask the question, why was there this initial suppression for the first 15 days, but then an increase in methane production? And for that, the first thing that we looked at was the degradation of bromoform. And our results using GCMS analysis were that the bromoform was gone within about the first hour, and analogs such as dibromethane were gone within 4 hours. This is very consistent with the literature results where we know that bromoform is a very volatile compound, so we were not at all surprised. However, this does lead to the question of what is happening during this 15-day period that changes in these last 15 to 20 days. So to understand that better, we did a genetic analysis, including quantitation and qPCR. For quantitation, we found that bromoform causes a toxicity response in the microbes. This is consistent with literature where we know that bromoform as an halogenated compound is toxic. So to get a more clear lens on what's going on with methanogens, we did qPCR using a methanogen-specific probe. And from this, we found a slight upregulation towards the end of the experiment of methanogens, in addition to this initial spike in relative concentrations of methanogens. We don't have a great answer right now of why exactly this is happening, but it could be because non-methanogen microbes are more affected by the toxicity effect of bromoform than methanogens. And so therefore our next step will be to analyze the microbial community structure using genetic sequencing to see how the populations are shifting over time and how that would then impact methane production by mapping onto functional groups. So that will be the next very exciting step in our research. But what we already know is that bromoform initially reduces methanogenesis, but results in greater overall methane production. That bromoform is gone within an hour, and that dibromethane is gone within 4 hours, so we are not seeing a chemical response. Additionally, DNA shows a toxicity response to bromoform, so bromoform is toxic. There's a slight upregulation in methanogen expression, so bromoform is slightly more toxic to non-methanogens than it is to methanogens, while still being probably toxic to the methanogens as well. And then our next steps will be to analyze the microbial community structure to understand more why this is happening. How are those functional groups being affected? How is methanogenesis exactly being affected? How are different species of methanogens being affected? Overall, from this, we're beginning to understand how bromoform works as a methanogen suppressor, why it works, and hopefully our further research will help us understand better how we can implement this technology at a farm scale. Thank you so much, and I hope you have a great day.